So this morning's talk will be by Professor Terry Peters, and the last 10 minutes or so will be by his colleague, Mark Henkelman. So Terry, we're lucky enough that Terry is close to us. He's only about an hour and a quarter drive down in London, Ontario. He's at the in the imaging research,
Imaging Research Labs.
Right, Imaging Research Labs at Robarts Research Institute at the University of Western Ontario. He's a professor in, let me see if I can get this right, radiology and nuclear medicine, as well as a professor in medical biophysics. He's published over 150 papers. He's given over 130, over 131 invited presentations. He's really an influential person in medical imaging, at least from what I've experienced. His name shows up a lot. And just to, well, he was an organizer of the 2003 MICI, which was supposed to be in Toronto, and if you remember, that's when the SARS, Toronto got all this bad press for SARS, so he was one of the two people responsible for reorganizing all that and planting it 2 weeks later in Montreal, and that was a big success. Conference I enjoyed a lot. I was told by a colleague of his that he independently discovered Fourier domain CT reconstruction just shortly after it was discovered by Bracewell. So this colleague of ours says that Terry's a very, very bright man. And he said to me before, Terry said to me before that he spent his PhD living in the frequency domain. And I think I've been there too. So I present to you Dr. Terry Peters.
Thanks very much, Jeff, and also congratulations on organizing such a wonderful meeting. I think it's just the sort of thing that we need to get images together with other mathematicians and computer scientists, because there are lots of problems that we can solve. I want to give a bit of a historical overview of where medical imaging has come from and how we got to where we are now. Talk about some of the applications and challenges, and then, as Jeff said, that at the end I'll hand over to Mark Henkelman, who will talk about some specific challenges in small animal imaging. Some of this will overlap a little bit of what Paul Babin mentioned yesterday, so hopefully it'll be complementary and you'll reinforce some of the things that he said. So I'll talk a little bit about some of these mainstream imaging modalities. There are some others, but there's just not enough time to go into everything. So medical imaging, diagnostic imaging, has been X-rays for the longest time, since Röntgen invented X-rays in 1895, and that's actually an image of full-body image of Wilhelm Röntgen. You can see his car keys in his pockets. Well, I guess they're not his car keys at the time. And the nails in his boots. I'm not sure anyone has ever calculated the actual dose that he received with that image. But that sort of technology was the mainstay of diagnostic medical imaging until the 70s. And of course, the 70s brought computers, and we started getting CT scans. So in the early 70s, CT scans. Again, in the 70s, PET scanning and SPECT scanning as well. Computers brought us digital radiography, which not only had the advantage of getting rid of the film as the capture medium, but it also allowed you to do image processing on the image much more readily than you could with film. And it also gave the opportunity of things like subtraction angiography, where you can see the background has been subtracted, so you just see the blood vessels. And of course, in around 80, we had MRI coming on stream. So the last 2 or 3 decades have been incredibly exciting, and I consider myself to have been particularly fortunate in having gotten my foot in the door in medical imaging back in the late 60s and starting a PhD, which led to the career that I've followed for the last 30 years or so. So if we look at some of the technology that started it all, the typical X-ray system still looks exactly the same. The major difference with X-rays is we've gone from a cassette piece of photographic film was placed in between, sandwiched in between these 2 sheets of fluorescent material, which would glow when X-rays would hit them. The film would actually be exposed more by the light than the actual X-rays. This has recently been replaced by an electronic receptor, which has about the same sort of form factor and can fit in the same sort of cassette holder that you would have on this X-ray system. But now we have typically 1, 2, or 3,000 by 3,000 detectors that can record the image. Digital radiographs now give us extraordinarily high-resolution images, high dynamic range, and as Dr. Babin pointed out yesterday, there's tremendous possibilities for processing these images to bring out subtle details that cannot be captured on film. And an example from my colleague Martin Yaffe in Toronto, with this breast image, you can see this region here, which has been enhanced by some image enhancement algorithm to really bring out the characteristics of this suspected tumor. Angiography, as I mentioned earlier, is the technique that is used to image blood vessels in the body, or in this particular case it's a CM angiography system that's specifically designed for the brain. And so you can have an X-ray source up here, a receptor down here, the patient goes in the middle, and you can orientate that in any direction. And this particular geometry can also be adapted for what we call cone beam CT scanning. And I'll talk a bit more about cone beam CT as well. Anytime you have the opportunity of collecting multiple projections of an object, you have the opportunity of reconstructing it into a 3-dimensional volume. So the angiographic information that comes from these sorts of systems is, as you see on the right, the cardiac angiogram, where you can see the contrast filling up the vessels around the heart. On the left, you can see the arteries. A few seconds later, you see the veins in the head. If you subtract these two from each other, you can see a beautiful picture of the arteries and the veins in the same image. And as we did a number of years ago, we made these images stereoscopically so that the surgeon could actually see a 3-dimensional view of the whole cerebral vascular tree. And this was extremely useful for planning trajectories to targets, to record electrophysiology information about epilepsy, for example, or Parkinson's disease, to try and put probes into the deep into the brain without passing through any of the blood vessels. Of course, we're all familiar with today's state-of-the-art CT scanners. Instead of giving you projections of objects, they give you cross-sections of objects. And as I mentioned, if we can record projections of slices or volumes from many, many directions, we can mathematically reconstruct them to form a 3-dimensional image. Inside a CT scanner, we have a source, X-ray tube that rotates around sometimes up to 3 times a second, an array of detectors, historically, this was a single array of 1,000 to 2,000 detectors. Now it's up to 64 of these arrays stacked side by side, so that you can scan a slab with 64 slices in it, that thick, in less than half a second. And so by spiraling this thing through, you can record large chunks of the body in just a few seconds. Just to emphasize the fact that there's more to image processing than just taking the final images and performing some sort of filtering algorithm on them, detection algorithm, multi-scale process. We also have access to the raw data. And in CT scanning, the raw data are these projections, and we can convolve them with this filter function to give us this very much high frequency enhanced version of this projection, and then back project this into the volume, and after we've back projected all of these projections, we get the reconstructed image. We can use the Fourier version of this convolution by taking the Fourier transform of this, multiplying the Fourier transform by this frequency enhancing so-called ramp filter, inverse Fourier transform, and do the same thing. This is mathematically equivalent to filling up the two-dimensional Fourier transform. If we take these projections, this one here, take the Fourier transforms, these happen to be the samples along the 2D Fourier transform of the actual image. So alternatively, we can perform these FTs, fill up this two-dimensional Fourier transform, and invert the two-dimensional Fourier transform. So there are opportunities of doing image processing at this level, in addition to doing image processing at the level of the reconstructed image. An example of what's actually happening when you're acquiring the data. This is showing the angle of view rotating around. Let me just repeat that again. Down here we have all of these projections being built up, and on the right, this is the filtered version of the projections, and then it's back projecting those. And you can see as the back projection angle increased, the quality of the image increased until we got an image which is virtually indistinguishable from the original. If you look carefully, you can see a faint shadow here, which is just the difference between the reconstructed and the original, showing the error involved. We've come a long way. Back in the early 70s, we got an 80 by 80 image. It did have two slices. It had a fairly wide beam, and there were two detectors that would scan in parallel. So you could get two slices, and depending on the model of scanner, somewhat between 1 and 4 minutes acquisition time, and between 4 and 10 minutes to reconstruct each slice. Today we can get 64 slices simultaneously, acquire them in much less than a second, and we can reconstruct probably much better than a quarter of a second per slice. With the multi-detector CT scanners, we have the ability to collect data dynamically. The data you see here from the beating heart, it's done using a gating technique, so we're acquiring images that are sorted into bins so that we get a full set of projections over each phase of the cardiac cycle. If we surface render that, we get these extraordinary images that you can see here, which are amazing when it comes to planning heart surgery. Many of you are aware of the trend towards minimally invasive heart surgery or robotic heart surgery. To have these sorts of images that show exactly where the vessels are, exactly where the occlusions are, is a major advance. Of course, we can also render the dynamic images to give us extremely realistic reconstructions of the beating heart. some of the angiography that was performed just with X-rays can now be performed with CT scanners. And so if you look at the difference between the image that you get with the cardiac angiography that I showed before compared with the CT angio, this is achieved by injecting contrast into the heart vessels and making sure you collect all of these scans while the contrast is still within the vessels before it's being washed out. And up here we see these extraordinary images of the carotid arteries and related arteries around the feeding the brain. Similar to the image that Dr. Babin showed yesterday, if we process these images in the appropriate manner, we can reconstruct them as if you had placed a camera inside, in this case, inside the colon. And so you're doing a fly-through of the colon. And as he mentioned, this is a lot more pleasant than having to suffer the indignities of a colonoscope. Well, I'm told that you have to be filled up with air before this happens, so I'm not sure that that's all that comfortable either. But you can see how we can display this information in a variety of different manners. In this case, you see the view that an endoscope would actually see. So this is looking at a polyp. But you can also take this data and fold it out into a strip and look at it's as if you'd taken the entire colon out, slit it along the middle, folded it out on the table and examined it. So it gives you a tremendous amount of flexibility in the way that we can analyze these images. Let's talk a little bit about magnetic resonance imaging. Some of us can remember when diagnostic MRI was actually called NMR, but then the nasty nuclear word caused its name to be changed to MRI, and it's been MRI ever since. NMR is still generally refers to the small magnets that you see in chemistry and physics labs. And the techniques of MRI all have been borrowed from the fundamental techniques that were developed in these chemistry and physics labs. How does MRI work? Well, here's a two-slide physics lecture. Take some protons, put them in a magnetic field, irradiate them with radio frequencies, and they start wobbling in phase. Wobbling nuclei in phase generate radio waves. The frequency with which they wobble depends on the strength of the magnetic field. So if we can tailor the magnetic field, we can get a bunch of different frequencies of wobbling. If we measure the frequencies of those radio signals, we can map out the entire three-dimensional distribution of these wobbling nuclei. So, no field, these nuclei are pointing in all directions. Put them inside a magnetic field, irradiate them, they start wobbling. Put a field gradient on, different frequencies come out, record them. Of course, you've got a bunch of different frequencies, you have to perform a Fourier transform somewhere along the line, you get the image. Another little example of how the MR image is actually made up from its frequency components. And this is the really cool thing about MRI. The raw data are actually samples of Fourier space. And I think that because of my history of swimming in Fourier space when I was a graduate student, the whole field of MRI and CT being so Fourier based has always held a tremendous fascination for me. So there's what the raw data look like, and this is what we see. If we are collecting the data from in an MR scanner, we're actually building up Fourier space line by line, or sometimes it's in spirals, or sometimes it's in lines through the origin. But in any case, every few milliseconds we're picking up a new sample in Fourier space. So the data collection part of an MR scanner is collecting these points in Fourier space, and then the reconstruction phase kicks in. You feed this data set into an FFT algorithm, and you go from here to here. Again, we can, through gating techniques, get the patient to hold the breath and put an ECG monitor on, gate the times at which you're acquiring the data. You can collect enough data to give you a full three-dimensional image of the beating heart. Magnetic resonance angiography is another major area where MRI has shone. And we'll see that MRI is not really a single modality. It's magnetic resonance, T1 imaging, T2 imaging, T1 weighted imaging, T2 weighted imaging, diffusion weighted, functional imaging, angiography, and it goes on. The cool thing about angiography is that, at least in principle, you don't have to even use contrast agent. It gets better if you use contrast agent, but because you've got so much flexibility in tailoring the pulse sequences, you can actually tailor an MR pulse sequence so that it only reacts to tissue that's moving. The background stuff just doesn't show up. So you can get extraordinarily high quality images like this without the injection of any contrast agent. Functional MRI, again, is another amazing application. Because the nuclei behave differently in different surroundings, it only takes a small amount of difference in the environment to change the way in which the nuclei, after they've been excited, recover back to equilibrium. And if we look at the rate at which the signal decays, we can see it's different depending on how much oxygen there is in the blood. Functionally active parts of the blood recruit more blood with more oxygen, and these subtle differences can be detected by an MR scanner when it's switched into functional MR mode. And so we use this so-called blood oxygen level dependent, or BOLD effect, that we then register. We get an image using this technique, register this to an anatomical image, and we can get a map showing the activity superimposed on parts of the anatomy. So this is just one of many, many examples of how this sort of technique is used, in this case to look at the difference in brain activity between a non-drinker and an alcoholic. Not quite sure what this says, whether it says that it's easier to do math problems when you're drunk, but I mean, hey. Often this type of technique is used for planning surgical techniques. You often want to get to this tumor, but you want to make sure that you spare aliquot tissue in the brain, so that you might, in the preparatory phase to a neurosurgical procedure, you might have the patient perform a number of tasks that would identify exactly where the motor and speech functions, for example, lie on his brain. You register that with a 3D image that shows the tumor as well, and then you know which parts that you should avoid. One fascinating image that was produced by one of my colleagues at the Robarts a number of years ago was this demonstration of ocular dominance columns in the visual cortex using fMRI. This was the subject of Hubel and Wiesel's Nobel Prize many years ago, when they actually discovered these columns, and they were using nuclear labeling techniques on rats, I believe, that had been subject to these stimuli. They'd been fed a compound that was appropriately labeled, and then they sectioned these brains and looked at the photographic film that was exposed by this radioactive label. We can now do this with fMRI. in vivo, which is really quite extraordinary. Another very fascinating application is diffusion-weighted MRI. Who would have guessed that the grayscale image that we're all quite familiar with could actually contain, or be coaxed to contain, information about the direction of fibers within those slices. But again, by manipulation of the pulse sequence and the gradients, we can attune the MR scanner to be sensitive to very, very small flow along nerve fibers. And so instead of the image being just a simple grey level image, it's a grey level image which has these little vectors superimposed on it. So if you could think of a stack of these images with the vectors, and we connect the vectors up, we can get amazing images that look just like they're from an anatomy textbook. And if you compare this with a picture in Gray's Anatomy, for example, they look almost identical. And the nice thing about it is we can actually collect all the data in a relatively short time. Of course, there are limitations because it's sensitive to very small movements. We've got to make sure the patient is very still. And this also is a potential problem for fMRI. And it's unfortunate that a lot of the patients, a lot of the subjects for whom many of these techniques would be very useful are the sickest patients who probably can't stay in the magnet for long enough, or they can't stay particularly still. So 30 years of MRI, we've come a long way. First MR brain scan image. It's almost worse than the first CT image, I think, but it really shows promise. And I remember the very first MR image I saw. It was by Lauterbur, who recently won the Nobel Prize for his work in MRI. And he was demonstrating at a CT meeting, actually, his latest technique called zergometography. And he had this picture of two test tubes of water that he reconstructed somehow using magnetic fields. This was way out in left field for all the people who were in the audience, who were really gung-ho on the latest CT techniques. But it didn't take very long for that simple concept. And I think the point of his talk was using magnetic field gradients to encode position, just in the way I demonstrated, rapidly blossomed into the sort of technique that can produce these images. 20 years ago, it took somewhere between half and 2 minutes to record data for a single slice. Today we can get sub-second imaging per slice, we can get volume imaging. It still takes about 30 minutes to collect all the data we need for a relatively high resolution beating heart image, because it means the patient has to hold their breath for each sequence. He has to hold the breath at the same level. So it's still complex to get good 3D beating heart images, but it can be done. Another technique that has actually been around for quite a while, and it's only been in the last, at least in Canada, 10 years where it's really started to be installed and be used. Positron emission tomography works by injecting a metabolically active positron-emitting isotope. Positrons meet electrons, they annihilate, generate 511 keV gamma rays. Set of detectors that record these. And once you've recorded that sort of data, you've got the data in the same form that we would have for a CT reconstruction. So just graphically, positron, electron, momentarily form positronium, and then annihilate, give us the gamma rays. And these gamma rays are detected by opposing detectors in the ring detector like this. We get many, many coincidences. The electronics detect events that occur within a several nanosecond wide window, and say that, and record the fact that if this detector and this detector get a pulse at the same time, then there must have been the gamma rays emitted 180 degrees apart from somewhere along this line. And we do this for many millions of lines, and we get the same sort of equations that we have for CT. Of course, these need to be interpreted in the context of the anatomy, and so up until recently, PET scans were almost always registered to MR or CT after the fact. Now it's becoming quite common. In fact, most PET scanners now, I think, are delivered with a built-in CT scanner, which means that the CT and PET are intrinsically registered. And so this not only provides the anatomy, it also, having the CT image registered to the PET data, also helps tremendously in the accuracy of the reconstruction, because you can use the information in the CT scanner as part of the PET reconstruction algorithm. So, for example, looking at this cardiac image, cardiac PET image, would mean a lot less if we just showed the colored part of it without the overlying anatomy. And again, this point was made quite elegantly by Dr. Babin yesterday. Over the last few years, and Mark's going to be talking a bit more about this later, 3D animal CT and MRI have really become a major research area, not only in the research to build these scanners, but research to analyze the images that come from these scanners. And they're particularly helpful to drug companies, for example. Developing new drugs, they want to see how these new drugs change animals. So scan animals at very high resolution, detect very subtle changes as a response for drugs. A small company that spun out of our labs at the Robarts a number of years ago was bought up by General Electric and is now the producer of this GE animal CT scanner, and GE actually makes these in London. Look at the extraordinary quality of image that you can get. So we've scaled down the size without sacrificing any of the resolution. This is a vertebrae of a mouse. This is the joint between in his knee. And these are not new. I think these were performed for David Holdsworth's PhD thesis about 15 years ago. Ultrasound is another amazing technique that is becoming more and more refined. Ultrasound transducer simply looks like this. It sends out high-frequency bursts of sound that get reflected back from organs inside the body. In the heart, for example, you might be scanning through the ribs, looking at these, a particular cross-section. And the neat thing about ultrasound is it's cheap and it's real-time. Showing some of the advances over the last couple of decades. Quite crude images, then 3D images at 4 volumes a second, 16 volumes a second, and now we can even see the blood flow in the chambers of the baby's heart with ultrasound. Aren't they cute? Ultrasound technology has gone miniature as well. This is an ultrasound image taken from inside the beating heart. The ultrasound transducer that produces that is no bigger than a match head. And it's fed either through the vascular system into the heart chamber, or even placed through a little opening in the side of the heart to look at this image. And what you're seeing here is a tool that's being manipulated around inside the heart chamber. Of course, these images make much more sense if we can integrate them with MRI. And as part of the work I'm doing in my lab at the moment, we're integrating real-time ultrasound with preoperative MRI of the heart so that we can use the preoperative image as a context to help us interpret what's going on at the time of surgery. And one of my surgical colleagues is very interested in performing surgery inside the beating heart. So instead of ripping the chest apart, taking the heart out of the body, putting the patient on a heart-lung bypass machine, he wants to do it without any of these invasive techniques. So if we look at the different characteristics, and I won't spend any time on these. They're in your book. You can look at the characteristics of the different modalities. MRI doesn't need X-rays. That's a big plus. There are some pros and cons for each of the techniques. CT gives us exquisite images of bony structures. It gives us exquisite cardiac images. Dose is the big issue. How can we get rid of the high dose and still get this high resolution, high dynamic quality images? Ultrasound, it's inexpensive, it's portable. Ultrasound doesn't travel through bones, at least not well enough to get a signal from reflected back from what's behind it. It doesn't travel through air. You get these huge refractive index interfaces that behave like light and refractive index, same sort of physics. So we can't penetrate some organs because of these factors. Nevertheless, it's becoming more and more widely used, and if we can get it adjacent to right on the surface of any organ during surgery, then we can manipulate it, and if we can track it, we can produce three-dimensional or four-dimensional images. One of the things we need is miniature four-dimensional transducers, miniature transducers that can be placed inside the body that can give us a dynamic three-dimensional image. Large systems like this exist, but the transducers are about that big, and they consist of an array of tiny little piezoelectric crystals that each get fired in particular phase relationships with each other and can actually steer the beam around in real time. Big problem, and this is going to be addressed by Mark as well, data storage. Where do we put it all? How do we deal with it? How do we deal with the throughput? How do we make sure we don't lose stuff? Dynamic cardiac scan, 5 gigabytes of data. So, what are some of the problems? We need to be able to visualize this stuff efficiently. How do we interact with the gigabyte data set efficiently? When patients are in the operating room or in the radiologist's office, he needs to give them an answer quickly. We need to be able to plan surgery quickly. We need to be able to combine these images with data from other modalities easily. We need to be able to employ different sorts of imaging visualization modalities in the same environment. Fortunately, one of the things that's helping us these days is the use of the hardware capabilities of, or the image processing capabilities of the hardware graphics boards. And thanks to the gaming industry, these boards are now relatively cheap but extremely powerful. Now this kind of looks humorous, but in fact, this is what neurosurgeons use. This is a bit of a segue into some applications that I just want to quickly look at. But how does a neurosurgeon navigate his way around the deep brain in order to place a stimulator to control Parkinson's disease? He sticks needles in it and sends currents in there and asks the patient what he feels, or looks and see what the patient does. And sometimes it's a knee-jerk. So that's the sort of, it's the data that are collected during that sort of stimulation process that can be used to build up a database of electrophysiologic points that we can map into a standard brain space and then remap back to individual patients. So this slide, this video clip is showing two things. One is showing the results of multiple stimulations on multiple patients, all having been mapped to an individual new patient. But it's also showing a visualization environment that combines three different sorts of visualization methods. Volume rendering over here, texture mapping of the original data. It's very important to, whatever you do, to keep the original data and be able to refer to it, cross-reference to it. And also surface rendering, the little blobs you saw in the middle. So it's important to be able to utilize all of these different visualization techniques. Image morphing is something that we do a lot, particularly in the neuro area and more often now in the cardiac area. A movement started in Montreal a number of years ago was to develop a standardized brain image to which data from multiple patients could be mapped. So they developed, and we've extended the non-rigid registration processes that we need to do this. So we can take a brain from one individual and non-rigidly warp it to a brain of another individual. This used to take 8 hours. We've got it down to just a few minutes now. So this can be used for mapping the electrophysiology data from individual patients into the standard database. It can also be used for doing the reverse, mapping the database to new patients. But it can also be used for taking an atlas that's been defined on the basis of the standard brain, mapping that to an individual patient. And this shows an example of the results of this, where we've got in the bottom panel the standard brain, in the top panel the actual patient that's being operated. You can see a representation of the surgical instrument, of the probes that are used to do the electrophysiological stimulation or recording, and representations of all of the different studies that have been performed on other patients prior to this new patient. And this data in this environment helps the surgeon to localize the point that he wants to stimulate or ablate during a new procedure. Again, another example of image fusion, preoperative MRI in gray, the intraoperative 3D ultrasound in brown, orange. You can see tumor shown in MRI, the same borders of the tumor, the borders of the same tumor shown in ultrasound. And this is demonstrating very vividly that brain moves when you make a craniotomy. So you can't just go blindly and guide your surgery based on what you saw in a craniotomy. But if you can acquire data about the state of the actual brain during the surgical procedure, then we can use that to correct the preoperative MR image so that it matches to the intraoperative ultrasound. So what are the major challenges that I see? Well, there's some quite pedestrian challenges that prevent some of the stuff from moving forward as fast as it maybe could. Having all of the 3D and 4D data sets available instantaneously to radiologists and surgeons in the operating room or the viewing room. Cross-referencing of images from one study to another study that might have happened a year, 2 years ago. Getting the same images automatically lined up. And exactly these points were made by Dr. Babin yesterday. Again, how do we rapidly interrogate a gigabyte of information? Zero into the parts that are of interest. And then seamlessly integrating all of these with a surgical guidance platform. And specific computational challenges, and I took these from a report that was recently commissioned by the Department of Energy and the National Institute for Biomedical Imaging and Biomedical Engineering. And these really gel with my view as well. Image fusion over multimodalities, multi-scales, resolutions, temporal, spatial. Extraction of display of quantitative information. And something that probably hasn't received nearly as much attention yet is not only data archiving, but how do we search those archives? How do we generate metadata that describes what's in the image? So can we come up with systems that will take all of the images and figure out what's in the images, even if they can't figure out what the diagnosis is? Give a system a bunch of images and it can separate them into brain scans, heart scans, for example. An untapped area at this point. And just one example of my lab where I'm trying to build a virtual environment that will help cardiac surgeons perform these new, novel, minimally invasive procedures in the heart. We're having to integrate preoperative images, map models to patients, integrate electrophysiology, ultrasound, haptics, endoscopic images. So there's lots of image fusion, lots of image registration, lots of image segmentation. So all of these areas are far from complete. Tons of research is going on in how to optimally segment structures, how to optimally register them. So all of these areas, visualizing all this data at the same time, all of these areas are ripe for research. So at this point, I'm going to hand over to Dr. Henkelman.
How many do we need?
Really want to make sure we hear you.
Thank you, Terry, for sharing a few minutes of your time, and for Jeff for allowing it. This is an unabashed commercial. If you normally skip commercials and go out for something to eat, I'll understand you don't have to stay. But I wanted to just point out that if you're going to develop sophisticated mathematical techniques for medical imaging, which is a very complicated process, you might be interested in playing in the field of biological imaging, which has some real advantages in terms of big data sets, some important questions. And so I thought you really ought to get a chance to see a bit of that today. It's an MR image of a mouse. Started from the front. Just working our way back. Here's the brain. In a moment you'll see the paws appear here. The ears are folded back. I've never learned the anatomy of the head and neck, but pretty soon we get down into the chest. Here you see the two chambers of the heart here. You see the lungs. You see the ribs wrapping around. Break through the diaphragm into the liver. The liver fills the whole anatomy. This is breakfast. The intestinal tract. You don't have to pump air into it. You could do a fly through the colon if you wished. You can follow the spine on the way out. Pretty soon we get down to the east end of a westbound mouse. This is a big data set. It's about 500 by 500 by 2,000 slices. I've only shown you about 1 in 5 slices on the way through, and sits at about a gigabyte. It's actually more detailed information than we're used to getting in human medical imaging. So it's a gorgeous sort of data set to play with. So we've developed a high-field mouse imaging center in Toronto at 7 Tesla, and when we were thinking about doing it, we said we can't really do 2 hours per mouse and be serious about biology. And so we built one that allowed us to do multiple mice at the same time. So these mice share the same gradient and the same magnet, but they have individual spectrometers on each one for doing the MR image. And that lets us get blocks of comparable and standardized data. Ulf was talking yesterday about the advantages of having data that's really standardized. And this is different than human data, which is always one of a kind. It's partly handwork. Here we tend to do groups of individuals, and gives us better data to work with. If we go to parts, we can do even better. Here's an isotropic brain image of a mouse at 32 microns resolution. It's almost down to microscopic scale, and we've just started to work with these data blocks, and they're gorgeous, and we're having fun with them. Whoops. How do I go back? Up and down buttons. Okay, that's better. This is a real question in mouse phenotyping. It's a really simple question. Which picture on this page is different? So we start with a mouse that's out of a genetically inbred strain. We have a large anatomical data set. We have another mouse that's come from some genetic change, either a random mutation, a known knockout gene, a knock-in gene. The geneticists can do fantastic things. And then the anatomical question, an imaging question is this mouse different than this mouse? And the thing is, I don't want to know about the posture or the color of the skin. I just want to know about significant anatomical differences. And that's a tough math problem, but I think an amenable math problem and an exceedingly important one. Have to do registration to get rid of the postural differences. We've done a bit of that in the brain so far. Here are brain images, slices out of a three-dimensional data set that have been registered into an average of 10 individuals. And we get, in some ways, a much better image. It's signal averaged, but it's averaged over multiple individuals that are genetically the same. And so certain structures come out even sharper in the averaged image. But there are some structures, like these small spots in this region of the brain, which are not under tight genetic control. They're either vascular trees or fiber bundles, and they tend to get averaged out. This is an odd thing, this average image. It's a map of the parts of the anatomy that are under tight genetic control, and it's suppressed the variable anatomy that changes from individual to individual. One of the most amazing things we've discovered so far is to take an average atlas like that, and then at each point in the atlas ask, where did the individuals that contribute to it have to move from to get to be part of the average? And this is a color overlay that describes that. And while you see some extreme regions, the olfactory bulb and the brainstem that have actually moved significant parts of a millimeter, over most of the brain, the average displacements are about 1 to 200 microns. And this is a huge advantage over working in humans, because humans are genetically all different. Genetically identical mice are cookie cutter the same. And so if you want to look for differences, you're looking against a very tight variance rather than a very large variance to start out with. Here's just a test to see how well we can pick out differences. We took 3 different strains of mice, 9 individuals in each, threw them all into one average, and then asked, if we go to a point in the average atlas, this black point here, where did the different individual strains come from? And we see that the clustering within an inbred strain is tight and tighter than the differences between strains. So this is a very expensive way to tell the strain of mice. You can also look at one of them's black and one of them's white. And this is just some segmentation where we have a known knockout mutant. We've segmented the brain on the atlas only so we can backpropagate it to the individuals and begin to ask what are the sizes of various brain elements. And here's a case where we have a homozygous wild type, a heterozygous, that's one of the alleles changed, or a homozygous mutant, and we see changes that can be picked up quantitatively. And that's really a mathematical approach to genetic phenotyping. Just as in clinical imaging, you don't get by with just MR. You have to use some other things. Here's CT of a mouse kidney. Now the resolutions are way down. These little spots out in the periphery are the glomeruli that do the filtration. They're about 30 microns in size. We're not quite down to the capillaries where we can join everything up, but getting close to that. And here's a typical genetics experiment. This is a wild-type mouse lung vascular tree. This is a mutant in the Notch2 signaling pathway, which is one of the developmental structural pathways. And this is a really neat math question. How are these 2 trees different? They're not identical. I mean, we can make multiple examples of each. How are they different in a statistical sense? And then more importantly, what can you do with the inverse problem? These things are built out of some kind of rule set. We don't know any of the rules. Could you look at these patterns of trees and work back in an inverse way and figure out what got changed in the rule set, which is really what you want to know about what happened with that notch gene. That's just a math question, but it's, or a math approach to what's a biological question. So in summary, I'll outline some mathematical opportunities, I think, in mouse imaging. High quality and high definition data to play with. I mean, it's gorgeous data sets. There's tight genetic control of the anatomy, so one's starting with known things and working to something else. There are experiments with large N, so if you want to see multiple replicates of the same thing, you can just make them. I think there are some new statistics needed for dealing with these. We've begun to find some of these deformation maps. We look for significant regions, different regions. We're not sure what kind of statistics to use. These are 10 to the 8th experiments happening simultaneously, 10 to the 8th voxels. I think we need some new statistical approaches. I think this is an area where mathematicians can play directly in biology. You don't have to go through medicine, you don't have to go through intermediates. If you really want to solve some of these problems, you can make fundamental contributions to modern biological science working almost totally within mathematics. And I think that's quite attractive. And finally, it's a very popular area, so there seem to be lots of research dollars associated, which never hurts. It doesn't drive things. And finally, it's an area that I think is a lot of fun. Thank you.